Last time we had a look at the structure of nerves. Let's have a look today at the way that nerves are transmitted. So let's see what we know on how it actually works. We have to remember that when we have a nerve, it basically only works in one direction. So, talking to some students, it's a question of whether and how nerves work. Basically, a nerve sends an electrical signal in one direction. Now, how does it do that? And the next one is, can it send multiple signals? So that's what I want to try and address now, is to work out that the fact that we can actually have perhaps more than one signal travelling up a nerve, but they would be well spaced out, and the fact that a nerve signal will propagate in only one direction. So to understand how this works, we've got to have some idea about what's going on inside this bit here, the nerve membrane. So let's try and draw this out. Now, a nerve membrane typically is one of these double membranes with these phospholipid bilayer, and what we're going to have in here are a variety of different proteins, and these different proteins are going to control how things move in and out of the cell. So let's look at this bit, and I'm going to choose to look at an axon membrane. Now, this axon membrane is normally what we would call at a resting potential. Oh dear. So we've got this thing called a resting potential. What is a resting potential? Well, this is basically when a neuron is not basically transmitting. So this is our state of play when everything is okay. And what we have in here, we've got basically an outside and an inside, and what we have control all this. We have ions, maybe positive ions, and we may have negative ions, and these are separated by this phospholipid bilayer with a few proteins in it, and this is an insulator. And we know that electricity is basically the movement of charged particles, and what we're looking at for this nerve impulse is a movement of these charged particles, and when this does so, then we get what's called an action potential. We get something that's sort of doing something. So let's deal first of all with the resting potential. So what we've got here is a state, the outside of the membrane is more positively charged than the inside. The membrane is said to be polarized, and if we can measure this, it is about -70 millivolts. That's the difference between the two. So the resting potential occurs basically as a result of the movement of sodium and potassium ions, so Na ions and K plus ions, and these are moved across the axon membrane. The phospholipid bilayer prevents these ions from diffusing across the membrane, and so we have to use special proteins, and we'll call these channel proteins. Some of these channels are described as being gated. In other words, they must be opened to allow specific ions to pass through them. Other channels stay open all the time, so we might have some channel proteins that stay open all the time. Let's put the membrane in here. And this is going to stay open all the time, which will allow ions to move in one particular direction. Now, how do they do this? Well, if we look at this sort of better, we can see that we've got here a hydrophilic part, and that means that this part of the protein is going to be hydrophilic as well. We've got this area which is hydrophobic, and again this bit's going to be hydrophilic again, and that is how the protein stays in this membrane. But if we look at what's going on here inside this little bit of the membrane, this is also charged, and we know that all the different amino acids can have different charges on them, and by arranging these, then positive ions are going to be attracted through this channel. So we've got some channels that are open all the time, and this allows sodium and potassium ions to diffuse through them. So they can always diffuse out, but they can't necessarily diffuse in. So let's create a resting potential. What do we need to do this? Well, we've got our sodium ions, and these can be inside, and they are going to be actively transported to the outside, whereas potassium ions, these are actively transported to the inside of the axon by here specific what we call intrinsic proteins known as the sodium potassium pump. But this pump, it's not equal. For every 3 sodiums that are pumped out, only 2 potassiums are pumped in. So it's unequal. As a result, there are more sodium ions on the outside than there are potassium ions on the inside. Therefore, basically, the sodium ions want to diffuse back into the axon down, this time, at what we call the electrochemical gradient. This is sort of given the name we give to a concentration gradient of ions. Whereas potassium ions will want to try and diffuse out of the axon. However, most of the gated sodium ions channels, they're closed, and that prevents the movement of sodium ions, whereas many potassium ion channels are open, which allows the potassium ions to diffuse out of the axon. This means there are more positively charged ions outside than inside, and this creates our resting potential. So we've also got, basically then, let's put them here. So my gated channels here are closed, but we do have some diffusion. So we've got some potassium channels are open. So what does this mean? Well, let's draw out our little membrane. If we have this as our outside, and we've got here our inside, we've got some sodium channels. These are pumping the sodium out, and we've got some potassium ones going in. We've also got a channel here which is going to allow potassium ions to move out. So this largely gives us a very positive on the outside and a relatively negative on the inside. As I said, this is about 70 millivolts here, comparing the inside with the outside. And this is how a nerve is resting. It's just sitting there waiting to fire. So let's have a look at what's going to happen now when we get something called an action potential. So this is basically some sort of stimulus comes along, and this is going to affect some sort of sensory sort of receptor. This sensory receptor, somehow the stimulus is going to be able to temporarily reverse the charges on the axon membrane. As a result, what we've got is the inside and outside rapidly changing, and we've got now plus 40 millivolts, and this change from our minus 70 millivolts to this 40 we call depolarization. A sudden change in the potential difference from what was negative to positive. So we've got our outside, we've got our inside, and what we're seeing here is some sort of massive influx of positive here against the negatives. As the impulse passes, then this membrane then basically tries to reset itself, and the neuron will return to its resting potential. So an axon here has a resting potential. When something happens, this action potential occurs, the protein channels in the axon membrane change shape, and it allows these ions to move across the membrane, and we get here basically a change in voltage. So the change in shape gives us a change. So these gated channels have got a special name in that they're called voltage gated channels, or voltage gated ion channels. So how does this all work? Well, we could do this by having a look at perhaps some sort of graph of this. Now what I'm going to have here is this is 0 millivolts. Let's go up here to plus 40 millivolts, and down the bottom here let's go to about 70 millivolts, and we're resting. So this is our, if you like, resting all along here. Now something happens, and what we see is an influx of chemicals, and then they're pumped out, and we return back to where we were. So let's give this a sort of a name here. So we're back there to our resting potential, and this here is our action potential. To go along here, to get up to this stage, we've got the process of depolarization. But once the action potential has happened, then we have a period of time when we have a repolarization, and it typically overshoots and then resets itself. So let's have a look then. Our neuron's got a resting potential, it's not transmitting an impulse. Some of the potassium ion channels are open. These are not the voltage gated ones, and we know our sodium voltage gated ones, they're shut. The energy of the stimulus here triggers some here sodium voltage gated ion channels to open. So let's work out what's going to happen here. So I can sort of identify some of these bits, I suppose. Let's do that. So let's say this is number 1. So this is our resting potential. We know that we've got here the gated channels, they're shut, but we've got the potassium ion channels open, allowing the potassium ions to move out. Now something happens here. This energy basically is going to open these gated sodium channels. And the ions are going to rush in. As well as doing this, the membrane becomes more permeable to sodium ions. So as well as having my little membrane and my gated protein has now opened up, so my gate's opened if you like, somehow this membrane has also changed, and also sodium ions can flood in through this membrane. So we've got this membrane has become more permeable to Na+ entry. Now what's going to happen here? Well, this is going to make the inside less negative. This change in charge is going to cause more sodium ion channels to open. So here we're getting stuff going on, so let's do this as number 3. And what's happening here is we're getting positive feedback. Positive feedback loops are not common in the body, not really, and we see most of these in nerves being sort of done. This is the main use of positive feedback, and this simply means that a little change is going to suddenly give way to a lot of change. So a little bit starts and then suddenly an awful lot. Now when we reach here, number 4, this is our maximum. So when we reach 4, this is our maximum potential difference. This is now at plus 40 millivolts. Something else happens. This is where our voltage gated sodium channels they close. And our voltage gated potassium ion ones, they open. Sodium ions can no longer enter the axon. But now the membrane is now more permeable to potassium ions. These potassium ions now they start to diffuse out. Now this, of course, is going to reduce the charge, resulting in the inside becoming more negative than the outside. Basically, we're going to get down to here. It's going to become even more. Initially, lots of these potassium ions diffuse out, and this results in the inside of the axon becoming more negative. In fact, it's more negative than its normal resting state. So this bit here is called hyperpolarization. The voltage gated channel proteins that are moving the potassium now close. The sodium-potassium pump causes sodium ions to move out of the cell. So now we've got an NaK+ pump, and that is going to move sodium ions out, and it's going to move the potassium ions in. And after a few moments we reach stage 7 up here, where we simply reach our resting potential. And at this point we could fire that nerve again. So that's our resting potential and our action potential. Let's see how basically this is going to move along and produce some sort of signal going in one direction. So what we need to try and do is to make sure that we move from basically one end of a neuron to the other, and you want it to go in one direction. You don't want it to sort of suddenly take a piece of nerve, depolarize this and send the signal in both directions. That is certainly what we don't want. What we need is our signal that's all happened here to go this way and not to be able to go the other way. How does this work? So the initial stimulus causes a change in the sensory receptor, which triggered this action potential. So that first part of the axon membrane is depolarized, and this acts as a stimulus for depolarization of the next region of the membrane. But once something has fired, basically we're looking at here some sort of resetting process. And while this is resetting, this is fired, this piece is now resetting, while this one fires, and this one's firing, while then this one starts resetting. So by the time this one is now fully reset, it's got nothing next door to it to try and tell it to fire. So let's see if we can try and explain what's going on here. So let's look at this membrane and let's think what's happening. I've got sodium ions on the outside, I've got potassium ions on the inside. This is effectively quite positive, and this is effectively more negative. This is our resting potential. Now, a stimulus comes along. As we get our stimulus coming along, we're going to see some changes. And the change we're now going to see is basically we're getting our sodium ions moving in, and this is starting to change to positive. Nothing's happened here. This is the next, if you like, little bit, and this is still negative, and this is still positive. But here we're seeing it become more negative and more positive. Now this is going to move along, so that what we've got now is still this positive here. This part of the cell is still at a resting potential, but we've got this action potential here. We've got still quite a bit of potassium in here still, and we've got this large negative outside. But what's going to start to happen is that this bit is starting to basically recover and start to push out. The potassium here. So let's look at what's going on. My potential has moved along. I've got a process where my K is going out. I've got quite a lot of potassium out here. This is where I've got negative this side. This still bit here is still resting. And we're starting to get here this system now starting to repolarize. So that a few moments later Here we're back at our resting potential. This is the bit that was heavily depolarized. This is the bit where my sodium ions, they're moving out, and this is the bit where they are readjusting. And this bit here simply takes some time to happen. This is given a name, and this is called the refractory period. And this is the time during which basically we can't send an impulse. And this is what prevents the propagation of the action potential backwards along the axon, as well as sending it forward. So we get something which is unidirectional. Now, how do we try and improve the speed at which these nerves move? We talked about two different types of nerve last time. The sorts of nerves we talked about either are just little nerve fibers like this, or we can have nerve fibers with Schwann cells around them. These are just myelin sheaths of insulation around them, and here we've got our node of Ranvier. So what have we got here? Well, basically, if we look at one of these, each one of these makes sort of a localized circuit. Basically, these myelinated axons transfer electrical impulses much faster than non-myelinated ones. This is because the depolarization of the axon can only occur at these nodes of Ranvier. So what we're seeing is the action potential jumping from one of these to the next. So instead of gradually moving along, it jumps. And this means that the speed at which this moves is a lot faster. And since we know that this is sort of requires repolarization, that requires a pump, that requires ATP, this is going to use less. So these are in fact far more efficient. So this will have propagation going along very slowly. This will jump from one to the other, which will make it considerably faster. Two other factors, of course, affect the speed. We've got one thing that affects it is going to be the axon diameter. The faster the impulse is going to be travelling down, the larger axon. So the larger the diameter, the faster the movement. This is because basically there's less resistance to flow of ions in the cytoplasm than in a smaller axon. And the other one that's going to make a difference is our old friend temperature. This is going to give things like mammals a terrific advantage over animals that are poikilothermic. So we think of this nerve system as an all or nothing system. I put a stimulus in. If it's large enough to turn a resting potential to an action potential, then it works. If the resting potential doesn't do that, then nothing happens. A certain level is required, and we'll call this the threshold level. And once we get past this threshold level, then we get this positive feedback. And in that case, we're going to get this system working. I hope that's given you an idea of the way that nerves work. This all or nothing. And we have a resting potential where cells just sit around waiting for something to happen, and then this action potential of a all or nothing process. And once we've got this all or nothing in nerves that control fingers, movement, myelinated ones, then basically we can send the signal really quickly by jumping by these nodes of Ranvier. If you've ever tried something, you can try and move your finger fast. Try moving it even faster, and even faster than that, and you reach a limit where you cannot move your finger any faster, and that is because you can't send the next signal down the nerve until that refractory period is over. So you can only move some things just so fast in your body, however hard you want to try. And in this way you can have several signals moving down one nerve, one after the other, following at a, if you like, discrete distance, and they'll move at the same speed. So yeah, we can have that. And that means that nerves can work in one of two ways. We can either send signals such that we send down the nerve one signal after the other. So at this end, this relay neuron here is working on how many signals are coming in, or we can have several parallel nerves coming in, sending messages. And this gives rise to different types of systems you can have in the body. That we can either have these which are sending signals one after the other, and if you're getting a certain number of signals in a time, then this is going to fire that relay. Or maybe we're going to have this other system whereby you're only going to fire this relay if you get signals from nerves 1, 2 and 3. There we are. How we try to transmit information. Next time we'll have a look at the synapse and what we can do there, and we'll look at that one next week when we do some more A-level biology, topic by topic. Take care. I'll see you then. Bye bye.